And from their expertise, they understand where to make these assignments, and they can make basically what's a reference atlas. This is a more detailed map. Our scientists then use this to go back to another piece of that tissue and do what's called laser scanning microdissection. So the technician takes the instructions, they scribe along a place there, and then the laser actually cuts. You can see the blue dot there cutting, and that tissue falls off. You can see on the microscope slide here, that's what's happening in real time. There's a container underneath that's collecting that tissue. We take that tissue, we purify the RNA out of it using some basic technology, and then we put a fluorescent tag on it. We take that tagged material and we put it onto something called a microarray. Now, this may look like a bunch of dots to you, but each one of these individual dots is actually a unique piece of the human genome that we spotted down on glass. This has roughly 60,000 elements on it, so we repeatedly measure various genes of the 25,000 genes in the genome. And when we take a sample and we hybridize it to it, we get a unique fingerprint, if you will, quantitatively of what genes are turned on in that sample. Now we do this over and over again. This process for any given brain, we're taking over 1,000 samples for each brain. This area shown here is an area called the hippocampus. It's involved in learning and memory, and it contributes to about 70 samples of those 1,000 samples. So each sample gives us about 50,000 data points with repeat measurements, 1,000 samples. So roughly we have 50 million data points for a given human brain. We've done, right now, 2 human brains worth of data. We've put all of that together into one thing, and I'll show you what that synthesis looks like. It's basically a large data set of information that's all freely available to any scientist around the world. They don't even have to log in to come use this tool, mine this data, find interesting things out with this. So here's the modalities that we put together. You'll start to recognize these things from what we've collected before. Here's the MR. It provides the framework. There's an operator side on the right that allows you to turn. It allows you to zoom in. It allows you to highlight individual structures. But most importantly, we're now mapping into this anatomic framework, which is a common framework for people to understand, where genes are turned on. So the red levels are where a gene is turned on to a great degree. Green is the sort of cool areas where it's not turned on. And each gene gives us a fingerprint. And remember that we've assayed all the 25,000 genes in the genome and have all of that data available. So what can scientists learn about this data? And we're just starting to look at this data ourselves. There's some basic things that you would want to understand. 2 great examples are drugs, Prozac and Wellbutrin. These are commonly prescribed antidepressants. Now remember, we're assaying genes. Genes send the instructions to make proteins. Proteins are targets for drugs. So drugs bind to proteins and either turn them off, etc. So if you want to understand the action of drugs, you want to understand how they're acting in the ways you want them to, and also in the ways you don't want them to, in the side effect profile, etc. You want to see where those genes are turned on. And for the first time, we can actually do that. We can do that in multiple individuals since we've